Hi, good afternoon, ladies and gentlemen. I hope you had a very fruitful session today. Um, we want to talk about uh, toxicology and safety as it relates to um, research and drug development. And yeah, so the teaching objectives for this particular lesson, on my perspective, I would like to introduce. And in the case of those who have done toxicology, perhaps to reintroduce the concept of toxicology, including the basic principles governing the science. And I would also like to highlight on some important basic toxicity studies which may be pertinent to drug discovery and development within our context. And then finally, to discuss some important issues to note with respect to toxicity testing of extracts, which a number of you are actually working on. Concerning the learning objectives, what you should be able to take out of this particular lesson, well, to start with, it would be good if by the end all delegates are able to define toxicology, whether it's your own definition or a dictionary definition, but something which comes out of an understanding of the basics. Then also to be able to discuss the definition of a poison as proffered by Paracelsus. We'll talk about this. We must be able to explain why toxicology studies are important for natural products, because sometimes you find that there is resistance from other people with regards to why toxicity studies should be done for herbs which have been used for a long period of time. And then also to be able to discuss the nature of toxicity, to state some important concepts with respect to toxicology, and to briefly describe the major preclinical safety evaluations that herbal products would likely require, and finally to state the toxicity studies that you as the delegates would need to think about in the work that you are doing. Okay, so let's start. So I want to start by a simple question, a reflective question, so to speak. And the question is, what is toxicology? What is toxicology? And if there is time, it would be good for the delegates to just discuss in pairs and come up with your own definition of what you believe toxicology is, and then share that definition with the class. So the word toxicology, it comes from two Greek words, toxikos and logos. Toxikos and logos. Toxikos being the poison which was smeared on arrows, and then logos has come largely to be defined as the study of. So basically, put simply, toxicology can be defined as the study of poisons, the study of poisons. But then the moment you bring in this definition, another question pops up. And the question that pops up is, okay, so what's a poison? And again, I always like for students to talk about this in pairs. And what this does is that it then deconstructs the whole idea of toxicology and everything else that we'll talk about, because we then go back to the actual foundations, to the actual basics, where we start thinking about these things. So ordinarily, I would want the students to discuss it and come up with their own definition of what a poison is. And if there is time, perhaps about 2 minutes or so, delegates can actually spend some time uh, discussing this and come up with a definition of what is a poison. So, having said what I've said before, there's a dictionary definition of a poison which I took off the net. And the dictionary definition of a poison is something like, it's a substance that is harmful or lethal to a living organism. A substance that is harmful or lethal to a living organism. I'm not sure how close your definitions were to this particular definition, but that's the common definition of what a poison is. And this definition is interesting because it's got the term living organism. And by the time you're talking of, when you talk of living organisms, this is something which goes beyond humans. But for the purposes of the work that we are talking about, that we are doing, and what we are talking about in this meeting, we'll probably restrict ourselves to human beings as living organisms. But this is more, it talks about plants as well as other living organisms. So with that definition, which says it's a substance which is harmful or lethal to a living organism, I would like us to think about this with regards to what is a poison. We'll start with hydroxychloroquine. My question is hydroxychloroquine a poison? I mean, most of you would remember hydroxychloroquine and how it rose back to fame during the COVID era as a drug which was said to be useful for prevention and or treatment of COVID. But of course, I mean, the clinical data is shown otherwise. But is hydroxychloroquine a poison? And if your answer is yes, yeah, it is, because we did get some cases of people being poisoned after taking this medicine inappropriately. Okay, my next question. And what of Coca-Cola? Is that a poison? Yes or no? You can discuss with the partner whether this is a poison or not. Okay, and hydrogen cyanide. And most of you probably say, yeah, obviously we know you take cyanide, you're gonna die. You're exposed to it, you'll die. But is it really a poison? And what about paraffin or kerosene? Is that a poison? And paracetamol. I have a headache, I take 2 paracetamol tablets, I'm fine. Is it a poison or is it not a poison? What about lemon juice or lemonade? Is that a poison? And what of water? Can you define water as a poison? And if your answer to all of them was yes, you're probably correct. And if it was no, you're also probably correct. What is a poison? So some Greek physician some years ago by the name of Paracelsus came up with this definition or this statement, which anyone who has ever done toxicology should be able to say in one paraphrase or another. He said, all substances are poisons. There is none which is not a poison. Then he said, the dose or the right dose differentiates poison and a remedy, or a poison from a remedy, or vice versa, however else you want to paraphrase it. And basically, what he was saying was, it's not really about the substance itself, but it has got more to do with the dose of the substance. So basically, what this statement then does or did was to then define all substances as poisons. Is water a poison? Yes, there's something called water intoxication. You take too much water, electrolytes become less concentrated, can mess up your system, and people have actually died from it. So back to our definitions. A poison, we defined it as a substance that is harmful or lethal to a living organism, according to the dictionary. And a medicine, on the other hand, is a substance which specifically promotes healing when ingested or consumed in some way. To account for Paracelsus's statement, a poison can generally, in colloquial language or just common language, be defined as any substance which, in relatively small quantities, can cause injurious effects to living organisms. And this is basically from colloquial language, from basic street language, so to speak. Now my question is, since medicines are generally used in small quantities and you don't really need truckloads of drugs in order for you to be poisoned, can we then term them as poisons? And when we teach students in pharmacy, we always tell them that medicines should generally be viewed as poisons. And the reason why is because you don't really need truckloads in order to cause toxicity to someone. So since medicines are generally used in milligram quantities, they should be viewed as poisons. And this is a very important point to note, even with regards to whatever compounds we might be working with, that yes, they might have therapeutic effects, but things can happen if they are taken in overdose and in other circumstances like that. I think for completion's sake, so that you can get an actual definition, toxicology, according to Kasrat and Doe, is defined as the study of injurious or adverse or harmful interactions between chemicals and physical agents. And in this case, physical agents, we are talking about radiation and living organisms or biological systems. So that's how broad toxicology is. So as a result, you'll find toxicology then traverses several fields and disciplines, including environmental science, occupational science, forensic science, etc., because of these interactions which occur between chemicals and living organisms or biological systems. Okay, so having said that, oftentimes when you talk to people who work with natural products, there are two statements which, when you mention the importance of toxicity testing, they will always bring out to you. The first statement is, if something is natural, then it is safe. And I'm sure you've heard that. And maybe some of you have actually even used the statement to say, okay, it is from plant origins, therefore it is bound to be much safer than something which is synthesized in the lab. Okay. And then the second one, they say, if people have been using this plant or herb for years and there have been no reports of adverse effects, then what are you worried about? It's safe. Our people have been using this plant for years and years with no reported toxicological effects. At this point, I would really love for the delegates to discuss that point, those two statements. And, from your own assessment, are those fair enough assumptions? That, well, if it's natural, it's safe, and that, because people have been using it with no reported adverse effects, at least no reported acute toxicological effects, therefore the plant is safe. And then we can always hear your thoughts. But then moving right on. But well, firstly, I'm not sure if anyone recognizes those plants, for those who work with plants. On the right here, we've got Ricinus communis, and on the left here, we've got Abras precatorius. And interestingly, when you study in the literature, the most toxic substances known to men are actually of natural origin. And ricin, which has actually also been used, quite a fascinating compound, has been used in some political assassinations, is derived from the castor bean, which is Ricinus communist castor bean plant. And abrin, equally toxic, if not slightly more, is derived from Abras precatorius, the rosary bead or seed, whatever it is called. So it's not really true that if something is natural, then it's safe, because we know of natural things which are actually not safe. Even if you look at aflatoxins, which are produced by fungi, which would be found on peanuts and other grains, which have those are pretty toxic, and they are of natural origin. Secondly, the moment you see this, I think most of you would then think about thalidomide, isn't it? So the second thing that comes to mind is, well, just because something is not acutely toxic, it does not mean it's not toxic at all. Because you can give someone a herb now or your compound now, and there are no acute toxicological effects, which is fine. But then that does not mean that there are no effects which would happen in the long term. Because generally, cancers take a long time to develop. And even in the example which I gave to you, of the aflatoxins. Aflatoxins basically cause liver damage, and that's aflatoxins. They cause liver damage, liver cancer, which occurs not immediately, but you start to see it after some time. And on the other hand, you've got some drugs like thalidomide, which the mom can take, and the mom is pretty fine. There's nothing wrong with the mother. It even takes away the morning sickness, but the teratological effects are then seen later on. And these are things that we have to think about, even with the compounds that we have. To say, okay, I've given it to mice in an acute, as an acute dose. There are no effects, but does it mean that there are no effects, in terms of the teratology, in terms of mutagenicity or carcinogenicity occurring in the future? And then another thing is, unless the drugs or unless the herbs are being used in exactly the same manner that they are used traditionally, extraction methods that we use or the extraction processes that we use could actually change or alter the toxicological profile of the medicinal plants. For example, when we extract and we are doing perhaps an ethanolic extraction, what we're basically targeting by doing an ethanolic extraction are compounds which are more hydrophilic, water loving. So it means that we are, in essence, getting rid of the more hydrophobic substances. And if perhaps, peradventure, um, there was an interaction, some form of an antagonistic reaction, and we heard Martha talking about antagonism yesterday, some form of an antagonistic reaction with compounds which are found in the hydrophobic fraction that we threw away, together with the toxicological compounds which are found in the hydrophilic compound which we keep, meaning that at the end we've kept the toxic compounds and the compounds which would have antagonized the toxicological effects of the compounds which are now in the hydrophilic fraction, we've thrown them away, meaning therefore that we are introducing toxicity which would not have been introduced had the compound been used in the manner that it is used traditionally. I'm not sure if that is actually making sense. And sometimes, if we go down to actually isolating single compounds, by virtue of the fact that you've got this one compound which you have now probably concentrated, the isolated compound can actually also exhibit its own unique toxicological profile, which is different to, the compound being in quotes in partnership together with other compounds within your extract. So that's an important issue to think about. And then also, depending on the type of extraction method that you use and how you are able to get rid of the solvents, some of the compounds that may be used during the extraction process could actually introduce toxicological risks. I mean, methanol is very good for extracting hydrophilic compounds, but you actually have to make sure when you do your whether it's your rotary vapor evaporation or freeze drying that you've actually got rid of most of the methanol, because it's in itself toxic. And then the other issue is the plant itself might actually be safe, but then plants grow in the ground, in the soil, and the soil might be the one which is contaminated with heavy metals, with pesticides, etc. And sometimes just doing toxicological studies on your extract can actually start to, show these things and lead you to investigating more to see if there's any form of contamination. And in fact, most regulators would actually want you to test for heavy metals, um, as well as probably bioburden, microbial contamination. So those are the reasons. I mean, there is a need for toxicological and safety evaluations of herbal products and extracts as we are developing them for use in humans. And unless you are using it in exactly the same manner that the herbalist would use it, the moment that you just deviate from the protocol, then it sort of starts to complicate things a bit because of the issues which I mentioned above. Having said that, we just want to spend a few minutes looking at some basic principles in toxicology. I've just tried to take what I feel are the more important ones, and then I left out the rest, because probably we would not be able to finish this with the time allocated. So the first principle I want to what's called the nature of toxicity. It's important to realize that all toxicological responses are not the same. And even as you're doing your experimental work, this is something that you always want to keep at the back of your mind, to say, okay, what type of toxicological response is this? And they exhibit differently depending on a number of factors, some of which we'll talk about. So the nature of toxicity is therefore varied. And as we're doing our experiments, like I said, we need to always keep this at the back of our mind. So I shall go through some of the different toxicological responses that occur with compounds that we should also think about. Okay, so there are some responses which can be immediate and some which can be delayed. So some toxicological effects occur soon after exposure, but others take some time before they occur. The ones which occur soon after exposure are much easier to pick out because you've given your compound 5, 10, 15 minutes later, you actually see the effects or the adverse or toxicological effects of your compound in an animal if you're using an animal model. But the ones that take a bit of time may be a bit more difficult to catch, because once you maybe perhaps when you leave your animals in the cage and you go home, you come back tomorrow and you find that something has actually happened or the animal has actually died. So it's actually delayed. So with the compounds that we're working with, we need to always ask ourselves this question. Okay, I'm not seeing anything now, but is it because there's a delayed effect? Or perhaps there's no effect at all toxicologically? I hope that makes sense. On to the next one. We have some effects which are reversible and some which are irreversible. So toxicity, reversibility is a good thing. If you have a toxicological effect which is reversible, well, that's good, because then you know that after some time it will go away. And these are probably the ones that we would actually see as side effects, which occur, with the drugs that we take. So, for example, reversible effects could be maybe damage to the liver, which is not substantial, where there's regeneration, or even sedation. Sedation doesn't last forever. I heard during the lesson yesterday, when I think it was Robert or Martha, I don't remember now who or both of them, when they were talking about binding and how after a drug has bound to a receptor, we would expect it to leave so that the effect can be terminated. And when we're talking of reversibility, it's actually termination of whatever adverse effect or toxicological effect that we are seeing. The ones that we are more concerned about are the irreversible effects. And these ones are the ones that would cause permanent damage. For example, your CNS neurotoxins, or even your aflatoxins, if they are going to lead to liver cancer. And these are things that we should also think about intentionally as we're doing the work that we are doing, to say, is there any reversible toxicity? Is the toxicity irreversible or not? Psychologically, there are what are called local effects and what are called systemic effects. So here, I just put this because I like snakes. So this is a bite from one of the snakes that we have in Zimbabwe, in Southern Africa, called the stiletto snake or Bibron stiletto snake. And basically, the envenomation or the toxicity is just largely restricted to the bite site. But you can then have compounds which can cause toxicity to the heart, or which are more systemic, which go elsewhere. And you want to ask yourself these questions. The compound which I have, does it cause any effects which may be localized, or does it cause effects which are more widespread? So I just gave two examples here. Like if someone gets if hydrochloric acid spills onto your skin, the effects are largely localized. But if another acid, hydrofluoric acid, spills onto your skin, in addition to the localized effects, it is actually absorbed into your body, causing systemic effects as well. Then some effects are cellular compared to then others are molecular. So you can have compounds which are destroying the cells and cell structures, but compounds which go even much deeper, destroying DNA, which can ultimately lead to cancers and other things like that. Another important issue, which you might not be able to see perhaps in preclinical studies, is the issue of hypersensitivity. But it's also something that we just have to keep, at the back of our minds, that individuals respond differently to different chemicals or compounds. And a classical example in the drug world would be your sulfonamides or your penicillins, where there are people who are just really hyper allergic to penicillins or sulfonamides and can have serious issues like your Steven Johnson syndrome. So that's an important thing also, even as you are developing your compound, things to be thinking about at the back of it as well. And then also, I mean, there's the issue of inter-individual variation, that people are not the same. The way that they metabolize the medicines is not the same. The way that they would respond even to toxic, exposures is not the same. This one is of particular importance, interspecies variability, because all the animals will also not respond the same. And the classical example would be thalidomide, um, which in rats was found to be safe. It was later demonstrated that perhaps a rabbit model would have been a much better one. So even if we're still getting some, some good safety data from animals, which is really good. It's a good pointer. It's something also to keep at the back of our mind. And I think there's a slide where I'll talk slightly a bit more about this. So I guess we can leave it at that for now. So that's with toxicology or toxicity. The second thing, which is extremely important, is the issue of pharmacokinetics and how it influences toxicity. You see, poisoning or toxicological effects cannot happen unless a toxin or a toxic substance first enters the body in sufficient concentrations to cause toxicity, which means absorption becomes a very, very important issue to look at. And whilst I'm here on absorption, in case I forget to talk about it when we talk about interactions. I hope not. Okay, let me talk about it when we talk about interactions. And then the other issue is the issue of distribution. How does the compound distribute itself? Um, Here we're talking about the compound that you're using vis-a-vis maybe your positive controls. Are there any interactions that could occur? Um, and then also how long the compound remains bound in at the target organ or in the body before it's eliminated. So you'll find that when pharmaceutical companies want to bring a compound onto the market, they have to do ADME, your absorption, distribution, metabolism, and excretion tests, and must be able to demonstrate it. But when you're looking at your extracts, these are extremely difficult to demonstrate, and we'll talk about this probably in a later slide. But it's always important for you to ask yourself these questions, even as you're giving the compound that you're giving. You know, how is the absorption? How is the distribution like? What's the metabolism like? What's the excretion like? Is this compound influencing the metabolism in one way or another? Important concept that I want to talk about is the concept of target organ toxicity. So you'll find that compounds generally, from a toxicological perspective, seem to favor certain organs. Not all organs are affected equally by toxic agents. And due to a number of factors, like perfusion, maybe the receptors which are there, maybe, and other things like that, you'll find that the certain substances then tend to gravitate towards certain organs. And this is also important, that when you're dealing with your own compound, you ask yourself, okay, from what I know from the literature or from what I found out toxicologically, where would I expect this compound to have its effect? And that's an important thing to look at. So you'll find that I will not talk about safety pharmacology from a preclinical toxicology testing perspective, but in safety pharmacology, these are some of the things that you're looking at. which organs will your compound work on? And most of us are working with compounds which work on the CNS. So perhaps we need also to pay a bit of attention with regards to the central nervous system. The fourth concept is something which Martha talked about yesterday, and the concept of dose and response. And this is really key, or a foundational a concept in both pharmacology and in toxicology, to be able to see that relationship. So this is supposed to be a sigmoid curve, but as you can see, you just say it's dose, but here we know here it should be on a algorithmic scale from what you learned yesterday. So from a toxicological perspective, this is extremely important because you see what is written NOAL, which is the no observed adverse effect level, and LOAL, the low observed adverse effect levels. These are the ones which are then ultimately used to calculate doses that would be considered, in quotes, safe for first in human trials. And this is if you are dealing with compounds which have not been used in humans before. But for most extracts or compounds that we have, they probably have been used. But I guess that's something that we can also we'll quickly talk about in a later slide. And then there's a fifth concept which I just put here for completion's sake. There's something called hormesis in toxicology. I heard someone talking about heavy metals. I don't remember what heavy metal it was in particular. But you find, like, with trace elements in the body, when there's a deficiency of trace elements, the adverse effects are much higher. And people can actually die if they are not there. Then as you continue to increase the dose, you get to a point where you've got the region of homeostasis. This is the normal region. We want the body to be working around here. But when you continue increasing the dose, the response moves back up. So it forms a U-shaped curve whereby deficiency is bad for the body, but also an increased amount is also bad for the body. Also, something that you can think about with regards to the compounds that um, you might be dealing with. And I just put it here for completion's sake, in case somebody stumbles on some curve which looks something like this, which is not the normal sigmoid curve that we expect from the compounds that you might be dealing with. So I just want to quickly go through some of the pre-marketing safety evaluations which are necessary. From the talk which Martha gave yesterday in the afternoon, it was in the morning, I don't remember now the time. When a regulator is looking at medicine, there are three things generally that all regulators will invariably look for. And the first thing is really the issue to do with safety. And this is whether your compounds are synthetic or they are from a natural origin. The regulator wants to ensure that they are safe, that the public is guarded from the compounds. The second issue has to do with quality. And I heard Martha talk about it yesterday. When you say paracetamol is 500 milligrams, it should be 500 milligrams. And those are quality issues. And your batch of paracetamol, if you describe it as being white, we can't have a bottle with white paracetamol tablets, 50 of them, and then the other 30 are brownish, and then there are 2 which are black. But they must be of exactly the same quality. And then this is expected of all medicines, invariably. So even after you have gone through your preclinical work and you're developing your medicines or your compounds into drugs which you want to get market authorization, you need to ensure that there is proper quality. And this is the realm now of pharmaceutics, where the pharmaceutists would come in. Then there's the issue of efficacy. And for efficacy, all allopathic medicines are expected to satisfy this. And I am talking about your paracetamols, your aspirins, etc. When you send data to the regulator, they will look at this and see what do the randomized control clinical trials say with regards to the compound which you claim you want it to be used for, whatever it should be used for. And I think COVID, when it came, it showed us really the workings of the regulator, because you'd hear such statements as, phase 3 clinical trials are still underway and other things like that. But for complementary medicines, it's not really a necessity, because in most jurisdictions, for complementary medicines, what you need to demonstrate is that the medicine has been used um, traditionally over a period of time, and also you do not make any efficacy claims. So, for instance, here in Zimbabwe, if you have got a herbal product which you are bringing in as a complementary medicine, they will look at safety and quality. And then for efficacy, there is always a sticker which says something to the effect of no approved therapeutic uses, which means that there is no clinical data from randomized control clinical trials, that's what the RCT stands for, to demonstrate that your compound can actually be able to manage or treat a certain condition. And this is important for all those people who are doing research from a complementary medicines perspective. So we'll talk a bit more about this later. So basically, we said safety is key. There's an important statement which these two workers said. They say toxicity tests are not designed to demonstrate that a chemical is safe, but to characterize the toxic effects a chemical can produce. And that's so important, having seen what the definition of a poison is, isn't it? That you really want to characterize, you want to show that in the event that somebody takes this in overdose, what are the things, the toxicological effects that we are looking at. So when you look at the way that the FDA, as well as EMA and other competent regulatory authorities, would look at new chemicals coming into the market, or even the way that the pharmaceutical companies would develop their compounds from a toxicological perspective, it follows this pathway where there is test material identification and then characterization of the chemical, a literature review, structure activity assessments, and these are extremely important. And then it comes down to short-term animal studies, which could be and acute and short-term repeated toxicity studies, which could include or involve also in vitro genetic toxicology as side studies that you'd carry out in addition to this, as well as metabolism and pharmacokinetics. We talked about the issue of metabolism and pharmacokinetics earlier. And then subchronic toxicity studies. So here you're talking of studies that take between 3 to 6 months or thereabouts. And then they would look at reproductive chronic toxicity studies of more than 6 months and oncogenicity. That's basically what a full spectrum of toxicological studies would look like ordinarily, with also safety pharmacology here at the side. But for our purposes, because most of the times you're dealing with compounds which have already been used, and also the work that we're doing is still pretty preliminary, we would not expect, all these tests to be done, although it is possible to. So the type of safety evaluations which are commonly used are in silico studies, or computer tests, that's what I'll just call them, test tube studies or in vitro studies, in vivo studies, as well as clinical studies. And we'll just quickly go through these, which I'm sure you've been. So with your in silico studies, these would be your docking studies, where you are looking at the uh, chemical of the formula of the chemical structure of the compound of interest, and you do docking studies using computer models to see how these compounds would, in essence, bind. I've put that in quotes. It would bind to a receptor where other compounds with similar structures bind to. And this would then predict whether your compound will produce some toxicological effects or not. And then this is a question which I like to ask. So do you think that these would be useful for traditional medicines or for our extracts? Let's say you've just done a hydroethanolic extract of a compound of interest. Would docking studies be something that you would consider? Yeah. See, the problem now with the extracts that we have is that when you do your ethanolic extract, you've got probably hundreds of chemicals in there. or at least at the least tens or 20 or whatever, of compounds which are in there. And when you're doing docking studies, they work with one particular compound. And unless perhaps you have identified the pharmacologically active component of your extract, then it becomes extremely difficult to actually be able to use those. Although they could be useful, perhaps if you've isolated one single compound. So then in vitro studies, you normally use these ones, they provide you to find out information on mechanisms of action. Although you can also use in vivo studies for that as well. And they provide an early indication of potential for some kinds of toxic effects. So ordinarily, you'd want to start with in vitro studies, and then if your compound is too toxic at in vitro level, probably it's abandoned at that stage. So they're widely used for screening and ranking chemicals, for studying cell, tissue, and specific target specific effects. So these ones are the ones which are basically done outside the body. Examples from a toxicological perspective would be your cytotoxicity studies, which look at the potential of a compound to cause tissue damage, your protein binding, your cytochrome P450 inhibition induction, membrane permeability, as well as the others which are listed there. From this list, the mutagenicity one is a particularly important one, and I trust that I have a bit of time maybe to talk about. And then the advantages, well, they are largely less expensive to run compared to the in vivo ones, and the results generally come out much faster. Disadvantages, they are somewhat less predictive of toxicity in intact organs, because they do not account for pharmacokinetic issues. They do not account for how the drug will be absorbed from the stomach, or how it will probably move to the liver, whether it will be metabolized or not, what the distribution is going to be like, and how it will be excreted in an intact animal. And they do not account for whole animal reactions. And they cannot generally, predict, well, test neurobehavioral effects. Although, of course, I mean, you could do studies which are targeting particular receptor sites, which would give you some information about what to expect from a toxicological. And then moving on to our in vivo studies. And these ones are normally the ones which are most commonly used for risk assessments for regulatory purposes. And generally, for an in vivo toxicity study to be complete, you'd want to use one rodent species and one non-rodent species. And the types of studies here would be your acute toxicity studies, which are normally done, so it's normally within 24 hours. Your subacute toxicity studies, your subchronic toxicity studies, and your chronic toxicity studies. So subacute ordinarily would go up to 28 days, could even be 30 days. Chronic would be anything that is above 6 months. Then you do have lifetime studies for compounds which you expect someone to take for the rest of their life. So the type of in vivo study that you would actually use will be determined by what the drug you want to use, what the drug you are testing for will be used for ultimately. About acute toxicity studies, which I think should be done for um, all compounds, I think, in my opinion. So basically, these are generally the first toxicity, in vivo toxicity tests to be performed in a new chemical. And they estimate the LD50. If you remember LD50, although many people are now moving away from this concept of LD50, but LD50 is what you would use to calculate your therapeutic index. I'm not sure if we've talked about therapeutic index yet. We did talk about ED50 yesterday. So LD50 or therapeutic index would be the ratio of LD50 over your ED50. And it's supposed to just give you an indication of how toxic a compound is, so to speak, although strictly speaking, there's some issues with that. So they provide also information on the target organs. Remember, we mentioned that most compounds have got organs which they specifically target, as well as clinical manifestations. And it's also important to identify the species, any species differences, susceptible species, as well as to establish reversibility. We talked about the issue of reversibility. You want to basically stay away from compounds which have got toxicological effects which are irreversible, So these acute toxicity studies would actually be able to assist you. And they provide information for design and dose selection for longer term toxicological toxicological studies. Thing about in vivo studies. So basically, in vivo studies, for in vivo studies to be useful, or maybe the premise for in vivo studies is there are two main principles that underlie all descriptive in vivo animal toxicity studies. And the first one is that the effects produced by the compound in the animal are applicable to humans, predictive toxicity, toxicology. And this is basically what we also do for the neuropharmacological test that we do, isn't it? We assume that if we see an effect in the animal, this effect is probably going to be replicated in human beings. And that's the same type of mentality that would keep with the toxicological as well. And then the second issue is to do with the fact that we assume that exposure to high doses is a necessary and valid method for discovering possible hazards in human beings. All right, so just a bit about these animal models. Um, you know, so 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 there are some reasons why animal models may be inappropriate. So it's important to think about this with the compound that you are working with if you decide to do toxicity studies. So animal models may be inappropriate in cases where there's a lack of an appropriate drug target in the preclinical animal model. So you can actually do if your compound maybe affects a certain drug a target which is found in humans but not in the model that you're using, then that model becomes of no use. Also, if there is a presence of an irrelevant target in the animal which is not found in humans, and we do find some cancers developing in mice which do not actually develop in human beings because the mice have got certain targets which compounds can bind to, resulting in carcinogenesis or mutagenesis. And then also the issues of differences in the metabolism of the compound. The metabolic process that a compound may take in an animal may be different to that in human beings, as well as differences in susceptibility to infection, as well as other specific pathogens. So I just put this slide for completion so that we can always think about this to say, the compounds that we're dealing with, or the models that we're dealing with may not necessarily be the best models to use toxicologically. They may not translate to human beings. So just some food for thought. Okay. Then this question often comes, so okay, so why do we do these in vivo toxicological studies? And one of the reasons is they assist us to establish a safe starting dose for clinical studies. Without this, it would be practically impossible for us to be able to know what dose we would start to use in human beings for a compound that we have. And these toxicological studies start to give an indication. Remember, I mentioned NOAEL and LOAEL. So using those, you can actually do some calculations, put in some safety factors, and then predict what dose is most likely to be safe in human beings. And also provide information on a drug treatment regimen which should produce the least toxicity. I think yesterday in one of the lectures we saw the minimum effective dose and the maximum tolerated dose, and such studies would be able to assist us to come up with those. And then they also assist us to be able to assess target organ toxicity and its reversibility. We already talked about it, and may provide insight to biomarkers for clinical monitoring. For example, if someone is exposed to, I don't know, organophosphates, we know that pseudocholinesterase levels increase, and you can actually be able to get some of this information from preclinical in vivo studies before moving on to human beings. And then maybe just quickly that you also have subacute studies, which are repeated doses, and they used to give us information on chemicals that toxicity that may occur after a chemical is given on a repeated basis. For those who are doing studies, perhaps on Alzheimer's or dementia, which are conditions that someone would probably have for a long period of time, you'd need to do, this type of studies because you administer for a period of time. It's not like somebody just takes the drug once or twice like you would with paracetamol, but it's something that somebody needs to take for a period of time. And then maybe whilst I'm here, so for both acute and subacute toxicity studies, there are basically 3 or 4 domains that you want to look at. So after you've done your study, you want to first, well, during the study, you want to look at the clinical parameters. What is the general health of the animal? If it's acute toxicity studies, ordinarily it's one single dose or several doses within 24 hours. And then for the next coming 2 weeks, you are looking at the animal to see where they will die and other things like that. And afterwards, you want to take the blood to look for biochemistry results. And afterwards, you also want to take the major organs in the body, the liver, the lungs, and if sorry, the liver, the kidneys, maybe the GIT. And if you feel that there are some other organs that you want to take, you also want to take them for histopathological examination. And those are the main three that you would look at. Okay. Then whilst I'm here, I'm not sure whether I'm sure probably when during I think there was a topic on ethics, the issue of the 3 R's has become extremely important from an ethical perspective. That as much as possible, we want to, if possible, replace the animals that we use. So if there are other methods which can be used to replace use of animals, well, that's excellent. Would want to do that. But in cases where the methods we do not have methods to replace the animals, then we want to reduce as much as possible, the number of animals that we use. And then finally, the issue of refinement. We want to have methods which cause as little suffering to the animals as possible. And I thought this was an important thing just to put here. I think that my time has moved. I've been talking too much. And then just a quick thing, to give you an idea of how you translate animal toxicity data to first in human trials. And I mentioned this in passing, and I'll still mention it in passing, that you want to have first a good dose response curve, and then to be able to estimate your NOEL. So NOEL would be the highest dose at which you do not get any toxicological effect. So this is basically at response 0. So the NOEL here is 10, not this one. But there are cases where maybe the least dose that you used actually causes a toxicological response, in which case you've got the low, which is the lowest dose, which is associated with an adverse effect of the lowest observed adverse effect level. And these two are basically what I used. If NOEL is there, perfect. Then you use some safety factors to account for interspecies variability, to account for pharmacokinetics, as well as other issues, to then calculate a dose which you would use in human beings. I hope that makes a bit of sense. And then just a thing to note, that there are some toxicological effects which are hard to predict, in animals. For example, if there is headache, you can't really tell that an animal has got a headache. So you can actually miss that, and this will only be found when you come to the clinical studies in human beings. Yeah, just that there are some safety assessments do not finish with the animal models. But even when your compound does get onto the market, there are clinical trials that still, as you're going through, you identify more adverse effects. But the animal studies also continue to progress. So maybe let me not spend. So now the key question, perhaps, and where all what I was saying was leading to is this. Okay, so how are you expected then to proceed, for yourself? You've got a compound that you've been dealing with. From a toxicological perspective, what is it that you are supposed to do? And probably these are the most important slides. I should have maybe started with them, but I wanted all the background information to come out. So the most important place to start is the literature review. For most of us, we're dealing with compounds which have been used in our countries or in our localities for a long period of time. And you'll find that with most of these compounds, work has been done on them. And your first point of call, even before you consider doing in vivo studies, is to find out what is known about the compounds. Have any phytoconstituents been extracted? Has any toxicological work been done on those? And you will find that if work has actually been done and there's sufficient data to demonstrate safety in preclinical animal studies of the compound that you're dealing with, then it may not be necessary for you to do your own toxicity studies per se. And what you may need to do maybe is to look at the compounds that you're using in terms of heavy metal contamination, in terms of maybe contamination with other chemicals and other things like that. So literature review, very key. And if you've got a compound which will be extracted, are there any similar compounds which have been extracted in the same class? Are they alkaloids? Are they a specific type of alkaloids? And what is expected from them toxicologically? Do you expect the same things to occur with your compound toxicologically? And once you feel that they still need for some tests to be done, the next place would be for you to do acute toxicity studies. And these are extremely important for whatever compound you're dealing with, because we want to know, in the case that somebody is poisoned or they've taken your compound in overdose, what is it that we should expect? And these are very important also if you're going to proceed to do repeated dose toxicity studies, because they help you to determine what dose to start with. And also quite useful, like I said earlier, in identifying target organs of toxicity. And for those with an interest in vivo as well as in vitro toxicity studies, I would strongly suggest that you look for OECD guidelines, which are readily available online, which would define different types of acute toxicity studies, repeated dose acute toxicity studies, as well as in vitro and other toxicities. And then if your compound is going to be used over a period of time, it would also be important for you to also consider doing some repeat dose toxicity studies. And for most of us, we're dealing with compounds which have got some, neuropharmacological effects. So I would suggest that in the tox studies, you put in neurotoxicological parameters. And there's a great paper by Mosa and McDaniels, I think, which I forgot to put in my references, but I could share the link to that particular paper, which talks about how to do a good neurotoxicity study, looking at the various neurobehavioral and other neurological parameters that you could examine. Another important one, is to also just have a look at mutagenicity. Your compound might not be acutely toxic, and that herb might have been used for years and years, and there were no reports of acute toxicity. But what are the effects, in the long term with regards to the potential for cancer formation? And for those with the biochemistry background, I think you'd be familiar with the Ames test, which is something you could do at the very least to check if your isolated compounds have got mutagenic potential or not. I mentioned the importance of toxical pharmacokinetic studies, but well, if you're dealing with a crude extract, that's difficult, because unless you've got a pharmacological marker that you will follow through, which is also very difficult, because with these extracts, sometimes you get potentiation, sometimes you get synergistic effects, sometimes you get beneficial antagonistic effects occurring. So it's really difficult for us to come up with pharmacokinetic studies, and most regulators would not really insist on this for your if it's your herbal products or your extracts. Yeah. And then you might also want to think about these things. If you're thinking that your compound will be used in pregnancy, then you might also consider doing reproductive toxicity tests, looking at potential teratogenic effects as well as other parameters related to the reproductive system. And generally, for compounds which will be for long-term use, it's always a good idea for you to consider carcinogenic tests and lifetime studies. So, how do we proceed? The way you proceed really depends, in terms toxicologically, that is, on what you are using. If you are using an extract in exactly the same way that it is used traditionally, then what you want to try and do is to find out traditionally if there have been any adverse effects and other things like that. But generally, it's at this level. It's we expect that it is in courts. It makes more sense to look at your compound more favorably, if I can put it like that. But of course, with your extracts, so many other things come in, which, well, it's now beyond the scope of this particular lecture. And then if you're using an extraction method that more or less, you want to ask yourself, are you using an extraction method that more or less targets similar phytoconstituents? For example, if the local traditional healers, use water to extract their compound, and you use a hydroethanolic extract, then perhaps we can say, the regulator can look at that a bit favorably and say, okay, more or less you're extracting the same type of compounds. You're targeting the same type of phytoconstituencies. Then if you're using an isolated compound, um, that then complicates things because you want to know whether it has been extracted before. If not, then it's as though you are now coming up with a very totally new molecule. And what would be used by the pharmaceutical company in terms of coming up with drug development is also what you would probably also the direction that you would also follow. Yeah. And then well, yeah, I think I've already talked too much. Let me just end here. Yeah, so I hope it was a bit useful. I tried to bring in some practical insights for those who are working largely with herbs and herbal products. But I think, yeah. Well, thank you so much for listening to me. I think I've got one more lesson that I will give on metabolism and drug interactions. But until then, merci beaucoup. Have a great afternoon. We'll talk again later. God bless you, and I'm out.